Good evening. I'd like to welcome everyone to our final Boston Seminar Series event of this academic year, and we'll be hearing from Professor Jaenisch in a few minutes. I just want to begin by giving everyone a brief welcome on behalf of the MIT Club of Boston. And for those of you who are, well, I would normally say for those of you who are local to Boston, we have a lot of other events planned, but of course, now with the new normal, all of our events are virtual, so anyone can join us from anywhere. We have a book club coming up that will be on July 11th, that will be discussing The Three-Body Problem by Liu Cixin. Very good, really good sci-fi book if you haven't read it. And at the end of this month, on Thursday, June 25th, we will be having a webinar on the science of chocolate with Professor Michael Sima from the Department of Materials Science. And that is still open for registrations as well. You have to bring your own chocolate for the taste along portion of that. Also, I have to like to acknowledge our sponsors for the Boston Seminar, for the seminar series as a whole. And our partner for this is Charles Schwab. When Chuck Schwab founded his company more than 40 years ago, he set out to challenge the status quo with a radical idea, that a business was here to support the people it served. This idea has been foundational to everything that Schwab has done over those 4 decades. Even in changing and challenging times, the company's dedication to tirelessly grow and provide service to others still stands. Similar to how we encourage MIT alums to engage in lifelong intellectual and emotional partnerships with MIT, Schwab encourages all clients to be fully engaged with their finances and to own their tomorrow. So thank you, Schwab, for your partnership and support. And now, to introduce our speaker for the evening, Rudolf Jaenisch. He's a founding member of Whitehead Institute for Biomedical Research and a professor of biology. In fact, he was my professor of biology many years ago for 721. So now it's very exciting. I get to ask the questions tonight. He's a pioneer in the development of transgenic mice. We've worked with them a lot in the lab. There's very important for a lot of breakthroughs that have come since then. And cancer, neurological and connective tissue diseases, as well as developmental abnormalities. And Professor Jaenisch has explored basic questions such as the role of DNA modification, genomic imprinting, and X chromosome inactivation. Most recently, the lab has been focused on using the induced pluripotent stem cell, or IPS, system to study diseases such as Alzheimer's, Parkinson's disease, and autism. He's a member of the National Academy of Sciences, has been honored with the Boehringer Mannheim Molecular Bioanalytics Prize, the Peter Gruber Foundation Award in Genetics, the Robert Koch Prize for Excellence in Scientific Achievement, and the Otto Warburg Medal as well. We'd like to ask everyone to please, we're going to take questions at the end of his presentation. And if you have questions, please enter them in the Q&A box instead of the chat box. And we will be monitoring both throughout the presentation if anyone has any questions or any issues that come up. I guess that I will turn it over to Professor Jaenisch.
Okay, well, thank you, Megan. So good evening, everybody. I don't know, since I don't see anyone, so I don't know who am I talking to. But anyway, so what I would like to do is, and I share my screen now, let me see. I would like to talk about some issues we are particularly interested in, is stem cells, genome editing, and what I think how it will affect how we do perform medicine in the future. And to talk about medicine, I think, on the human medicine, but really it's useful to talk first, to put it in the perspective of evolution. So let's see what's happening now here. So evolution. So this is from it's a really nice museum in Paris, the grand evolution of how animals developed. And of course, that's the main story, and it goes upwards, but it may not go that much upwards. Anyway, what I want to start with is we have 22 pairs of autosomes, 2 sex chromosomes, 6 billion base pairs, and many variation. And that really determines whether we are large, body size, and so on and so forth. And why did I? So, okay. Our genes determine our phenotype, our eye color, whether we have 5 or 6 digits, for example, and whether we get cancer. It is believed that, so there are quite a lot of genetic diseases, and it's estimated that everybody of us carries deleterious genes. Often they're heterozygous, so we don't need, but the probability of developing is very high. So this is now the chromosomes packaged in the DNA, and this is the sequence, and mutations anywhere in the sequence can give rise to serious disease. For example, as an example, if you have this mutation, you have 100% Alzheimer's disease. If you have this mutation, there will be no consequence, and this one you might get cancer. So that's what I want to say. That's what our genes encode and what the phenotype would be. So there are different types of diseases that pose different challenges. There are monogenic diseases. These generally are rare. They have a clear genetics, and they often have a very robust phenotype. There are sporadic late onset diseases, and they are much more common. They're medically most relevant because they're frequent. They have no clear genetic basis, and they likely have a very subtle phenotype. And we want to study that using stem cells. So it's a complicated thing. The 2 technologies that will be important, which I will discuss, is embryonic stem cells and gene editing. They are really the foundation to make progress to understand these type of diseases. So this is early development. From a fertilized egg, you go to cleavage, and then you implant, and you get an embryo, and then you form an adult. You have adult stem cells, blood stem cells, connective stem cells, neural stem cells, and they form the mature tissues of the adult. And ES cells are derived from what's called the blastocyst. This is the embryo in humans, maybe 100 cells before it implants into the uterus. And under the right conditions, you can generate embryonic stem cells from those. So the embryonic stem cell technology has really revolutionized how we can study development and disease. And this gives you an example, a cartoon of a human blastocyst, where these white cells are the ones which form the embryo. To generate an ES cell, one digests away the outer cells, which form the placenta. And this puts in the tissue culture. And if things work out and you're lucky, these cells begin to divide. And then they divide in the dish, and then you can expose them to different factors, and they tend to differentiate into various cell types, all cell types of the body, pancreatic cells, liver cells, neurons, muscle cells, cardiomyocytes, and so on and so forth. Basically, every cell type of the body. And clearly, if you would have immunologically compatible transplants, it is thought that these cells could really be used for therapy of many diseases. So I will give you some examples. So this is the, again, the scheme. You start from a human blastocyst, you get embryonic stem cells, human, and you can generate all these differentiated tissues, basically every cell type of the body. So it's an attractive system to obtain mechanistic insight into human diseases and into develop therapies. And just more actual, what we are very interested, for example, to use this technology for SARS-CoV-2 research. And if I just give you in one slide what we're doing is, so the disease phenotype of this virus is, of course, lung, acute respiratory distress syndrome, which leads to often to lethality. In the brain, major problems to the brain like strokes, heart problems, kidney problems. It's a very, as you all know, a very complex disease. So one of the questions we're interested in, what is a key cell type for virus replication? Where does it have the virus to replicate, and what is the effect of virus infection to a given cell type? And the approach is to look at the virus tropism, and we're making now generate the relevant cell types from human yeast cells and expose them to virus, and we can measure virus replication, cellular response, and we can screen for small molecules that would interfere with virus propagation in a given cell type. So the embryonic stem cell technology is really the foundation to do this under very well-defined conditions. But I will not talk about this. I can just, yeast cells, induction of pluripotency, and the quest for eternal youth. It's really not a new concept. 500 years ago, this Lucas Cranach, this German painter, really had this born of the fountain of youth, where you get into one side as an old sick person, you get out on the other side as rejuvenated. So that's really an old concept. And the stem cells, of course, had revived this concept. And obviously we had lots of suggestions what this could be used for. Now the stem cell technology really has led to a lot of problems. Stem cell tourism, you get offers, you go into the website and you find a million entries of people that offer you stem cell therapy. Much of this is tourism and bogus. I'll give you one example from the Swiss clinic here. They treat everything from burnout syndrome, endocrine dysfunction, impotence, anything. And they're very imaginative because they're using actually a stem cell serum from a rare Swiss apple. It's a technology enabled from the cultivation of the Huttwiler Spätlaube. This is a rare apple. And this is allowed to really treat these diseases. And you pay for this. So this apple serum is pretty expensive. So the problem is, of course, this is all bogus and very dangerous. So the concept of personalized medicine would be the treatment options that are optimal for a specific ailment. In transplantation medicine means use your own cells. And that's what stem cells could do. And it's really based on cloning. So Dolly the sheep, first cloned mammal in the 90s, really was the impetus for using therapeutic cloning. And it really changed our concept. What I learned in school was a pluripotent embryonic cell really develops into a differentiated cell, and this is an irreversible process. What Dolly said, it's reversible. And what it really says that the nucleus of a differentiated cell can maintain the potential to generate an entire organism. And this led to the concept of therapeutic cloning. And this is a simple scheme here. If this is just a normal fertilized embryo, you can generate from this embryonic stem cells and you can make therapeutic tissue. But if you want to transplant this into a patient, it will be rejected because, of course, the embryo has a very different immunological makeup than the patient. They're not matched. But if you do this by cloning, take a nucleus from a patient and put the nucleus into an enucleated egg, you can make customized therapeutic tissue. And this will be, of course, accepted because these are the patient's own cells. So that's the concept of therapeutic cloning, very controversial in the 90s. And so we asked actually quite a while ago, does it work in the first place? So we used a mouse which has immune deficient by this lacking this gene. And these mice are totally immune incompetent. It's a similar disease in humans. And so what we did was to take a skin cell, took the nucleus from a skin cell, put it into an egg, a mouse egg from its own nucleus was removed, made a blastocyst, cloned yeast cells, corrected the gene by gene targeting, I will come to this, differentiated those to hematopoietic stem cells, put them back into the mouse, and the disease was cured. So therapeutic cloning works. But if this is a human, this is really not an option because you would have to use human eggs, and that is very controversial. So the question was, how can one do it without eggs? And this was the birth then of the iPS cells by Shinya Yamanaka, which really basically transformed the field. So let me just briefly with one slide tell you what an iPS cell is. iPS cells coming from adult cells like fibroblasts, you transduce them with these factors, the Yamanaka factors, and by some scheme, you can select them for these iPS cells called induced pluripotent stem cells. And when this was published, initially when Yamanaka proposed it, it was really, people didn't believe it. It was too simple. But then a year later, he proposed in 2006, and a year later, Yamanaka's group, my group, and a former student of mine, Konrad Ehlinger, who's not MGH, at the same time published this. It works and you can make mice. And this was then followed soon by iPS cells from humans. And this really led to an explosion in this field. So if I just summarize what iPS cells are, this is a culture of fibroblasts growing in a dish. You transduce these factors, these factors, and you make a mouse. This is pretty amazing if you think about it. A single skin cell organizes into a complex animal transplantation with these 4 factors. And that was the concept that was used for was to take cells from a patient, and you can now reprogram those in various ways. I don't want to go into this, and you can generate the desired cell type you want to study because it's defective in the patient. And you can model the disease in the culture dish. That is probably the most important application. And I'll give you an example. ALS, amyotrophic lateral sclerosis, a terrible disease. You can generate from patients now iPS cells and compare them with iPS cells from a normal person. And you can differentiate these iPS cells to motor neurons, which die in ALS disease. And what you see is when you do this, you find these motor neurons from the ALS iPS cells have a phenotype, they die, in contrast to the control ones. So you can ask the question, what's different with those? Do they have symptoms or do they have a phenotype which is resembling the disease? And this does. And now you can use screening for small molecules to ask the question, can you identify drugs which interfere with the degeneration of these neurons in culture? And this would be a first candidate for a therapeutic. And this is being done, and it looks very promising. So the iPS technology, if you have as a main patient, it's a very terrible disease, you make iPS cells, and now you can use it for disease modeling in a culture dish, as I just said, or for transplantation into for diseases which are suitable for cell transplantation. And this is being done, and actually the first clinical trials using iPS technology are underway. So that's a very exciting development. The problem with this, I will give you one problem. So this would be the scheme again, making iPS cells. You can make these disease relevant, let's say, neurons for ALS. But then the question is, what do you compare it to? What does you control? That's very important. And the problem in this whole thing, the crucial issue is that there are unpredictable differences between individual iPS cells because of different genetic background. And you ask the question, when you find a difference, is it really disease relevant or is it due to this variation? And one of the key things is you have to make cells which only differ at the one nucleotide which is mutated, which gives rise to the disease. And this can be done. You can take iPS cells which have a mutation, and you can correct the mutation. You make an isogenic pairs of cells which only differ at this one nucleotide which is disease causing. Or you can introduce this mutation into wild-type embryonic stem cells. The point is you make cells which only differ at this one disease-relevant alteration. So that's what we did. And I want to come to Parkinson as an example. So Parkinson, as you all know, is it's an awful disease, and there are familial Parkinson, which are less than 10%. If you have a familial Parkinson, for example, a mutation in alpha-synuclein, you will get 100% Parkinson early onset. And so we wanted to make these isogenic cells to study this. And so this would be the synuclein gene. There are 2 point mutations which give you this early onset. And we generated these point mutations by gene editing. And I don't want to go into details. So 1 point mutation here, the other one here. Both of those give rise to Parkinson. So we have these isogenic controls. And now what we can learn with that. And this was from a collaboration with Susan Lindquist's laboratory. And when we compared the mutant with the wild type neurons, which differed just as 1 nucleotide, we saw that the mutant neurons got much earlier pathogenic phenotype in culture, but more importantly, they were able to identify a small molecule which affected, it's the ubiquitin ligase, which reverses the pathological phenotype. So this was a direct candidate to develop further for a therapeutical application that is being done in the company. And the other application was using the same pair of cells to look for mechanistic insights into gene environmental interactions. Now, you know, Parkinson is caused by pesticides, farmers, and so we could expose these cells to pesticides in the culture dish, and when the cells carry this point mutation, they were much more susceptible to pathological consequences. So we learned quite a lot from these very well-defined situation. But the most important diseases are not the familiar ones, but the sporadic ones, idiopathic. That's really what is clinically much more important. And that's a combination of age, genetic susceptibility, environmental risk factors, for Alzheimer's, for example, often there's a positive family history. And to try to find genetic factors, one uses whole genome association studies to find really clues. So to identify risk loci in these what's called GWAS studies, whole genome association studies. So I give you an example. So sporadic idiopathic form of Parkinson. So all of us have risk factors in their genome, and they're protective factors. And we are exposed to different environmental factors. And we are either patients or we are not. So let's assume you have this combination of risk factors and protective factors, then you're a patient. If you have this combination, you're not. So the point I'm trying to make is, there are many, many genetic determinants in your genome which either protect or are a risk. And the combination of these interactions determine eventually whether you're a patient or which you're not a patient. So each of these factors has a very small effect size, and that's very important. So how can you study that? And so one approach is again by making isogenic cells. So you have now a control cell which has these genes. We don't know which one is a risk factor, which one is not. And here as a patient, we don't know which one the risk or not. And what we do is we just exchange those, the control for the risk and the risk for the control. Again, with the idea to make isogenic pairs of cells to study. And that worked very well for Parkinson. We could identify some of these low risk factors. But diseases are also dependent on environmental factors. So pesticides, physical exercise, what you eat, behavioral stress. We're exposed to this all through life. And this determines age-related diseases which really change your epigenetic makeup, either normal aging or age-related diseases. So the challenge is pretty big. So this is just a complex way of saying we have many, many factors which come together to make you a risk, and be exposed to different environmental stress factors. But all this is only relevant in one cell type, but not maybe in other cell types. So it's very, very complex. So how do we sort out genetic versus environmental factors? How can we study complex human diseases in the laboratory? What is a good experimental system to study human diseases? And how can we manipulate the human genome and the human epigenome, correcting genes? So that's what I would like to talk about. And again, coming back to this picture, there are point mutations, which you know, which cause disease or do not. They're silent. And the question is, possible to correct those mutations in human and treat the disease. And the key technology, of course, is gene editing and gene targeting. Now, historically, this was done in the 80s by what's called homologous recombination, where you have the genome here, you make a vector which has homology to the neighboring sequence for what you want to change. That you want to insert this sequence. And then by homologous recombination, you can do that. I don't want to go into details here. It's a rare event. So you do this in embryonic stem cells, and a rare cell, maybe 1 in a million, undergoes the right conversion. So you have to select this rare cell, 1 in a million, and then you can grow them up. You inject them into a mouse blastocyst, which I mentioned before, and plant this into a mouse, and you get what's called chimeras. So these cells now contribute to all tissues in the chimera, including the germline. So you can cross now this chimera with a white mouse, and then if you get white mice, you have converted basically this embryonic stem cells to a mouse, as I said before. So this is really works very well in mouse, yes. It requires strong selection. And it's very consuming. If you're good at this, it takes a year. If you're not so good, maybe longer, likely longer. The problem for human yes cells, it doesn't work. Works very inefficiently, but not totally clear why. So this gave then rise to the emergence of the CRISPR-Cas9 system. So let me just briefly introduce that system. You're probably more or less familiar with this. So the age of CRISPR is different from homologous recombination. It is a protein, Cas9, which is a nuclease, which can be bound to an RNA, to a small RNA, which can guide the Cas9 to a given position in the genome. You can synthesize these small RNAs very simply. And what it does, this RNA, it's guided to a sequence and makes in a very precise position a double strand break in the DNA. So there are two components, a guide RNA for homology search to find your target, and Cas9, a protein that cleaves the DNA. So that has revolutionized, again, biology and medicine. So this is, again, just giving you what happens when you make this double strand break and you then what happens, the cell has to repair it, and it does it by making a deletion or an insertion. It's an imperfect repair. In general, it leads to a mutation. Or if you add a DNA template, you can insert this template at the double strand break. That's by homologous recombination. So this can efficiently generate mutation and correct mutations. So it's like a molecular scissor. It cuts the DNA and you can manipulate this. And Hollywood hasn't escaped Hollywood's attention that this has enormous potential for, and even for movies. So how efficient is this technology for gene editing? And so I give you one example from our work. That was quite a while ago. We asked the question, when we want to target 5 different genes, multiple genes in an ES cell at the same time, how efficient is that? And 5 different genes, 50% of the clones we got had all 5 genes mutated. It was stunning, but still, if you want to make a mouse out of this, you need, again, going via chimeras takes a year. So the question was, could you do it directly into the embryo without selection? So we injected the guide RNAs for 2 genes plus the Cas9 into a fertilized egg and made mice. And it was stunning. 80% of the mice were homozygous mutations in both genes. I mean, when I saw these data first, I just didn't believe it. I forced people to do all sorts of experiments to confirm and they confirmed it. But still, we didn't know what type of mutation it was because it was either deletion or insertion. It was not targeted. So the question is, could you make a targeted mutation? For this, we injected the same into the embryo, in addition, 2 pieces of DNA, which had a point mutation, which we wanted to introduce into these genes. And the pups we got out of 60% of the pups had the predetermined point mutation in both genes. It was stunningly efficient. And so based on this, so just summarize what this CRISPR technology does. You inject CRISPR and the DNA into a blastocyst, you get a mouse. This whole thing is very rapid, it takes 3 weeks. That's the gestation period of mice. So we made these double mutations, which I had mentioned before. The conventional way, it took us 2 years, one after the other. This took us 3 weeks, just the gestation. So it's stunning. So is it useful for medicine? Gene therapy for severe diseases. And so there are 2 levels you can think about it. Somatic therapy, it's correction of a mutation in patient cells. This could be done in vivo or ex vivo, and it has consequences only for the patient. The other level would be germline therapy. So this would be correction of disease-causing mutation in germ cells or in sperm, egg, or embryo. And it has consequences for the next generation. So I will talk about these two applications to human medicine. First, about somatic therapy, correction of mutations in patient cells in vivo. And I want to talk about an example of Duchenne muscular dystrophy, which is a really terrible disease. It only affects boys. It's on the X chromosome, females are carriers, and it is always lethal. And you can see it, and you know, you lose all control of muscle. It's awful disease. And the gene is dystrophin, which is the biggest gene known. It's 2.5 million base pairs. Has many of these exons which have repeat sequences, and it's a very large gene. It takes 1 day to even transcribe the gene. And so there are many, many mutations in these exons. And so the protein encoded by the gene is called dystrophin. And this is a structural protein which is really stability to the muscle fibers. You see it in green here. It's a very important protein. So this would be a mouse which has its wild type, which all the dystrophin is in the muscle fibers. This would be which has a mutation in one of the exons. You can see there's no expression of this protein. And this mouse has a very severe phenotype, muscle phenotype. So now comes an experiment, which was, which I want to go into a little bit detail. So this, as I said, there are many, many exons in this gene. And for example, also in humans, exon 51 often have a stop codon. So the RNA stops here and you have a truncation and there's no protein. So what this group did, and I will come to this, what they did was they just deleted this exon, this CRISPR-Cas9. So what exon 4 would now directly splice into exon 52. And there would be no stop codon or you get a protein. It doesn't matter whether there are a few sequences are missing. So, So what they did is they put all these components, CRISPR and guide RNA into a viral vector and injected this into cells onto mice. And this was all work of Eric Olson, who is in Dallas. I think it's fantastic and fantastic experiment. So I give you the example of mice. So these are the mice I showed you. Whitehead mouse which have dystrophin, the mutant mouse. And now he deleted this exon which has a mutation. And we can see is now the mice re-express dystrophin. And 1 exon is missing, but it doesn't matter. Their function. And the mice really regain muscle function. It's a stunning result. That was done in vivo. And he has done it also in dogs. So dogs, there's a mutation in dogs, which is also a muscular dystrophy, and around again. So the point I'm trying to make is, these patients and controls, this technology allows you to, in principle, in vivo, to really think about an efficient therapy for a dreadful disease, a lethal disease. So let me come from genetics to epigenetics, briefly. So in genetics, we have mutations in the DNA. It's inherited. It goes to the germline. It's different in species. So I talked about this. Epigenetics is different. Epigenetics is the way the genes are packaged. They're packaged into histones, and they can be modified. That determines whether the gene is active or non-active. And the DNA can be modified, reversibly modified. I will come to this. So this step is actually crucial for gene expression. So there are 2 ways where gene expression can be altered, either by a mutation, or epigenetically, turning off the gene, turning on. This is really used in normal development. So epigenetics, I think, is a key mechanism how the environment talks with the genome, or the genome talks with the environment. So to make it very simple, what you ever had for dinner, in case you had dinner already, somehow had a very subtle effect on your epigenome, on your modification of DNA, very subtle. It has really disease relevance for cancer, very well established, for neurodegeneration, long latent diseases such as Alzheimer's, Parkinson's, and others, imprinting diseases, and aging, normal aging. But importantly, the changes in methylation are in principle reversible by life. So I have to define methylation in a minute. They're reversible, and this is used for therapy of cancer quite successfully. They're using drugs which change the epigenome, but these drugs are unspecific. And the question was, could changing the methylation state of specific endogenous loci? So let me define methylation. So this is cytosine, one of the 4 base pairs of the DNA. And this can be modified in this position by adding a methyl group. It's called the 5-methylcytosine. And this modification now, if that occurs in the genome, in the control regions of a gene, it silences a gene. So it's a very important epigenetic mechanism to turn on genes by removing the methyl group, or turn off genes by adding this methyl group. So basically, gene silencing is dependent on DNA methylation, on the histone modification, which are the proteins which package the DNA, and on nucleosome remodeling. I don't want to go into details here. So can you change it in a very specific way? And that is indeed possible by using CRISPR again. So in this case, one uses a CRISPR, a Cas9 protein, which is mutated. It cannot cut anymore. But it still can bind the guide RNA and get guided to the target locus. And now comes a trick. Demethylation occurs with some enzymes called TET enzymes, or de novo methylation with methyltransferases. What you can do is these enzymes can be fused to the Cas9 to form a fused protein, which now has a fusion to demethylation activity or methylation activity. And when the guide RNA gets to the target locus, you can either methylate it, unmethylated CpG, C, which is in green, or demethylated and methylated C, which is here in red. So this allows you to specifically change the methylation of a predetermined target. And this works very well. So this would be, again, this machinery you're using, and you can, for example, turn on genes in neurons, key genes, by just targeting TET, Cas9 to key protein, or you can use enhancers, or you can truly change the whole nuclear structure. I don't want to go into details. It's a really very, very powerful toolbox. So can you use this for specific disease loci? Of course, there's no change in sequence. It will be change in the methylation. So what diseases could we think about? We could think about cancer, imprinting diseases, or autism. And I will talk about autism as one example. So the example I will be is Fragile X. Fragile X syndrome is a very serious autism syndrome. It is the most common disability in males. It's again an X-linked gene, like the muscular dystrophy, it's on the X, and females are carriers. They don't get the disease. But males, if they have a mutant X, that's the only copy they have, so they get Fragile X. The cause for the disease is an expansion. It's an expansion disease. So let me explain what this is. The gene FMR1, here's the start codon, and upstream of this, there is normal people have a GGC repeat, 6 to 50, that's normal. But this can expand. And if it's more than 200, you have full-blown Fragile X. The expansion, really, the majority of mutations leading to Fragile X are these expansions, and they silence the gene. The DNA becomes hypermethylated, they repeat, and the promoter the histones change the acetylation state, and so on and so forth. So it's really a silence locus. So the question we asked is, so this schematically, the repeat, this is the promoter, both are methylated in black, these black lollipops, they're methylated, so the gene is totally off. The question is, if you not target the Cas9, demethylation enzyme, to the repeat, could you demethylate it to make them empty lollipops and also demethylate the promoter and turn on the gene? So then we did that. So this again is the repeat, methylated. This is the normal one, non-methylated. And so you target now again Cas9 to the repeat, Cas9 TET. And this is now the sequence. It would be a normal one, 20 repeats, the sequence surrounding it. And this would be a Fragile X patient cell, which is 450 repeats. They're all methylated. So the question is, when you know target, we made this target a single guide RNA to these repeats, and they would target there. And what's the consequence of this? And the consequence was dramatic. So this would be the repeat. It's 100% methylated in Fragile X. We got it totally demethylated in after editing. And importantly, also the promoter, which is upstream. It is fully methylated, black, these black dots here, and not active. And it got totally demethylated after editing, like in wild type. So that was really interesting. You target the repeat and you demethylate the repeat as well as the promoter. And the question was, does it make a difference? And it does. It turns on the gene to basically wild type levels. That's here by PCR, you can see this. So so if I summarize that, targeting of this repeat with Cas9 TET induces demethylation of repeat and the upstream promoter. It reactivates the FMR1 gene in yeast cells and in postmitotic neurons. And it rescues the mutant phenotype in postmitotic Fragile X neurons. It restores basically the normal electrophysiological properties of these Fragile X neurons. So that was very encouraging. And now the question is it really applicable for patient? Can we think about this? So let me give you my thoughts. Epigenetic editing is a developed therapeutic approach. I think we have to learn quite a lot still. For example, what is a fraction of cells that need to be edited for achieving therapeutic effects? We don't know that. For Fragile X, maybe 15%. Can we, and this, of course, related, how efficient can we do the transfer of genes into the brain? So there's a lot of progress here. The key problem is the disease condition reversible, also developmentally set? So many diseases, when they're wrong, the developmentally set, you probably cannot do much about it. For Fragile X, it is likely you can affect the phenotype even after in mature neurons. But there are many of these issues need to be resolved. So what are the applications of genome engineering? So for disease modeling, I gave you examples. Now we can make pigs, rodents, of course, monkeys, which define mutations, define mutants in the same gene as what humans have. Really good models. You can make the human yeast cells. I talked about this. And you can make targeted gene therapy. In vivo editing, I gave you the example on muscular dystrophy, and combined with cell therapy. Now, those approaches are in clinical trial, and they're very promising results. But the key question I want to come to is, should you do this with human embryos? Should you manipulate the human germline? So let me just come back to what you inject Cas9 and guide RNA into the blastocyst, and you get the mouse, and it's very efficient. There's no selection needed. That's why it works in embryos. And then it's rapid. In mice, it was 3 weeks. So should this be used for the human germline? And so we're coming now to germline therapy. It's a correction of disease-causing mutation in germ cells, and its consequences for the next generation. In contrast, somatic therapy has consequences only for the patient. So that, of course, makes it a much more complex issue. So what would one use it for? Correction of disease-causing mutations would be one possibility. Inactivation of susceptibility genes or expression of protective genes to achieve disease resistance, another, or enhancement. So I want to divide the discussion on two parts. First, the scientific issues. What scientifically can we say is that, how safe would this be? And then come to ethical issues. So one thing is very important. If you have a disease, now let's take a dominant disease like Huntington's chorea, where if you have the mutation in one gene, you get 100% Huntington's disease, a terrible disease. So if you wanted to correct this and have not embryos, correct the embryos, then you would embryos which are from a heterozygous father, 50% of the embryos would carry the mutation, and 50% would be wild type. And there's no way to distinguish those two. So if you do this manipulation, you can't distinguish the wild type embryos from the mutant embryos. So any manipulation will alter the genes in 50% of normal embryos. And if you have a recessive disease where both, then it's even 75%. I think that's a problem. So disease resistant by inactivation of susceptibility genes. So what people think is HIV receptor, and I come back to this, it has been done, to achieve resistance to AIDS, or correction of another gene which protects against heart disease. Now here, one has to think about alternatives. These diseases can be really corrected in somatic cells. You don't have to do it in embryos. So it may not be required. And finally, enhancement, an example would be inserting a growth hormone to make taller babies. And it certainly would work, but I think enhancement poses other than scientific issues. So, what are the stages at which genome editing could be used to modify the human germline? So at fertilization, coincident with intracytoplasmic sperm injection, in zygotes. And this all the principal problems of these approaches is the success of the manipulation cannot be assessed. The reason is, if you inject DNA into a zygote, as we did with the mice, the manipulation may take not place at the one cell stage, but at the some of the cells are modified, others not. So if you do pre-implantation genetic diagnosis to take one cell from the embryo and check, you don't know. You can't make a conclusion because it could be mosaic, could be wild type or the other cell. So that's really impossible. But if you do it postnatally, then you could do this. How about spermatogonial stem cells or oocyte stem cells in the cases exist? So let me come to this. The advantage would be these cells can be cloned. You can check success of a correct manipulation. So this is based on experiments done in the 90s by Ralph Prinster first. He took from the testes of male mice, he took spermatogonial stem cells and could show when he transplanted those into testes of another mouse, a white mouse and blue donor mouse, he could generate mice. Now the point is these cells can be grown in culture, they can be cloned. And this allows them opportunity for gene therapy. So you can manipulate a cell, clone it, make sibling clones, sequence, and be sure everything is fine. And then if it's fine, you can make a mouse. Or if it's for humans, you can make a human. And you can do it even for females. Now this is only done in mice yet, but from iPS cells, people have been able to generate oocytes which can be fertilized. This has not been done in humans, but in mouse. So if you do that, the use of spermatogonial or oocyte stem cells may resolve most of the scientific issues. The point is you can test what you did, right? So that's a very important point. So the scientific issues which I outlined before may be not impossible to overcome. So that brings me to the ethical issues. If you can solve the scientific issues, should this technology be applied to humans? Now, as you know, this guy, He, a Chinese researcher 2 years ago, did exactly that. He inactivated the HIV AIDS receptor, as I outlined before, in human embryos and implanted two blastocysts into a foster, and babies were born. Now, the experiments have not been really published. It's not clear whether it worked. People think it does, but it was an outcry in the world, and he's now he's jailed. He's in jail in China. But the consequence, of course, already there are companies, an example from China, who offer CRISPR-Cas targeted genome editing. So it has been shown it will be used. So that really raises the question, should we do that? Under what circumstances? So editing of the human germline, there are ethical worries. Mistargeting causes uncertain or unwanted problems. Spectre of eugenics, what can you use it for? The slippery slope to eugenics. Once introduced, it may not be reversible. Editing may exacerbate inequality. Arguments against germline manipulation. Intergenerational consent is not feasible. You cannot ask the next generation. It's impossible to predict the consequences. A threat to human dignity. Counterarguments. Intergenerational consent is not relevant for multiple other decisions regarding future generations. We don't ask our unborn next generation how they will cope with climate changes and so on and so forth. So this is a counterargument. It's impossible to predict consequences for other well-intentioned efforts to improve human condition. That's really what we know. And there's no shared conception regarding the notion of human dignity. So these are all arguments for and against. And this is a complicated issue. And the National Academy of Sciences 2 years ago had a committee to work out recommendation under which human germline genome editing should be considered. I was on this committee, and I will just summarize briefly what the committee concluded. So if there's no reasonable alternative to generate a genetically related slide, one should consider this. But the restriction to gene editing should be convincingly demonstrated to cause or to strongly predispose to these conditions. So it's very important to have very, very strong research. Availability of credible preclinical or clinical data. Maximum transparency consistent with the patient, and reliable oversight. So there was a very complex discussion, and obviously there were a lot of lots of worries about the application. So let me come to my last slide. Why would we consider that? And I mentioned many of those. Disease prevention, Huntington's disease, Tay-Sachs, cystic fibrosis, sickle cell anemia. If you could eliminate these mutations from generation, that might be a benefit. But the alternatives are, as I said before, would be pre-implantation genetic diagnosis. So you could select an embryo from a heterozygous parent. 50% of the embryos are normal. So you could select by pre-implantation diagnosis the embryos which are normal, which don't have a mutation, by taking one cell and only implant those. And this is the same for all the other diseases. And for many, sickle cell anemia, somatic therapy is available, which is being done. And the first clinical trials are promising. But there are clearly some situations where there's no alternative possible when both parents carry the mutation, homozygously in a recessive mutation, or when there's a homozygous Huntington's parent, 100% of the embryos will be affected. You only can have a genetically related child if you would correct it. So autosomal dominant ones, infertility on the Y chromosome. Those are some applications. Modifying disease risk, like HIV resistance. So that what was done by this guy here. I should say here, it is known if you knock out the receptor for HIV, you might get much more susceptible to a coronavirus infection. So if it worked, these two girls he manipulated may be at risk for coronavirus infection. Modifying risk genes which predispose you to Alzheimer's or to cancer. So there's also be other potential applications. Or enhancement. Can you make stronger muscularity, eye color, height, learning, memory? And of course, complex traits are much more challenging. So this people can think about to use it for. And we can look at the science here, but basically what is permissible, what's not permissible. This is not a scientific question. That society has to say that's what we are comfortable with, and that's we're not comfortable with. And that's of course a very complex, ongoing discussion where we set the limit. But the National Academy of Science Committee concluded that under very, very rare circumstances, one could consider this. So this is all I wanted to tell you, and I want to finish here. These are just some people who contribute to some experiments I did. So anyway, thank you for listening, and I'm happy to answer any questions.
Thanks, that was a lot, covered a lot of ground very quickly. So our first question, is anyone looking at corticobasal diseases?
Say it again.
Is anyone looking at corticobasal diseases?
Cortical base. So give me an example. What do you mean?
The question asker didn't give me an example.
So let me answer the question in principle. So these are all pretty serious diseases, neuronal diseases. And you can study these diseases, any of these diseases, using iPS cells in culture. Can you remedy them? Are they accessible to cell transplantation? That very much depends on the disease, and that may not be for these type of diseases. Can they be, are they accessible to gene therapy? Very complicated for these type of diseases, because to just get to these cells. So I would think that we are far away from that.
What are the implications of these technologies for the repair of lung damage in COVID-19 patients?
So that's a complicated question. So the lung damage really is caused by just destruction of some epithelial cells of the lung, but very likely by the immune erection. So macrophages begin to secrete cytokines, and they have all sorts of very detrimental consequences. So what really would be important to understand, what are the direct consequence of virus replication in a given cell? Can we then prevent that? And can we interfere with the immune reaction? So the immune reaction initially is very important to tone down the virus replication. But later, in those patients who become very sick, the immune reaction is very detrimental. So we really are trying to understand which cell type is key here. Also the brain. Why do people get strokes? Why do they get demented after some of these, as clinical data show? We are pretty ignorant now. Can the virus infect neurons? Can it infect what neurons? Can it infect glial cells? All these questions are, at this point, not easy to answer.
What do you think about, should there be routine genetic testing of potentially marrying couples to assure that they don't both have defective genes in the same locus?
If you know you're a carrier for a gene, for a recessive disease, I think it would be very useful to test embryos by pre-mutation genetic diagnosis to identify those embryos which got both alleles. So for sickle cell anemia, you could do that. And for many other diseases, for cystic fibrosis, and you select the embryos which are wild type in those you implant. Absolutely. But if you don't know whether you have a mutation in any gene, I think you don't know what to look for.
So to step away from genome editing in humans, a coworker of mine would like to know, is genome editing in plants also something that you've taken into consideration in the discussions of the ethics.
Yeah, so I mean, this will be, of course, very important to do is being done, gene editing in plants. And it will have major consequences. So the big issue is, now it's a totally different type of concept which comes here and becomes important. It's called gene drive. So gene drive, and I don't have really, it's complex. I don't want to go into this, but it's a modification which is based on CRISPR type of experiments, which can spread through the population very rapidly. And this has been done in flies and Drosophila. It's thought to be important to do in mosquitoes, for example, to kill mosquitoes, or make mosquitoes resistant to malaria, right? Because it would spread through the population. It doesn't need selection. And this, people think, can be done in plants. So you could make plants which are maybe resistant to some parasite or so, some fungus, without spraying the poisons. So that's what people think might be the application for this, but it's really, so far, it hasn't been really done using this type of technology. Although there are, of course, gene-modified plants, and gene-modified plants, of course, have a lot of people have worries about those. And many of those worries, I think, are because people really don't quite understand what was done.
Yes. Yes, and of course, well, yeah, and sometimes it can be used, think of like the golden rice, you can insert a gene that would be beneficial and adding a benefit to the, make a food functional.
Yeah.
Some people have talked about using that to, using it to produce drugs or something. Yes, we have a question of, yes, could genome editing also be considered for pathogen regulation, such as mosquitoes and malaria, which I think we've.
Yes, so I think, one model which I find is an interesting idea is, could we, so Lyme disease is particularly important here in New England. Go to the islands here in Massachusetts. I mean, 30% of the people got infected, so it's really it's very, very bad disease. And the host for the bacterium, it's really, of course, a tick, and the ticks get infected by biting mice. So the question is, could we generate mice which are resistant and cannot replicate the bacterium? And then the ticks would get infected, you could eliminate the gene. So the question is, can you make mice? And actually, there's a lab at MIT who's trying to do that. We are collaborating with them to modify using the gene drive concept, because then it would spread through the wild mouse population. It would make mice which are just cannot replicate the bacterium. It would not have terrible, unforeseen effects, I believe. So this would be one application. And when Kevin Esvelt, who promotes this, when he went to some islands here and talked with people and asked the inhabitants of an island, would they be willing to let, if he succeeds, to let free these modified mice, there was enthusiastic support for that. So, you know, there are potential applications. I think the more realistic application would be to spread resistant genes through mosquito populations, and they become resistant to malaria. And then one wouldn't have to spray these pesticides with all the bad effects.
One asks, can we generate modified T cells from iPS to create allogenic T cells?
Yes, that is really a very hot topic of research. It is particularly in cancer treatment. So what people do is, what they have shown is they can modify using CRISPR-Cas T cells to recognize an antigen which is just on a specific tumor. So this technology now is in the clinic. It is used for treating melanoma, some sorts of leukemia. It's extremely exciting. And the problem is you have to get cells which are matched. And what people now do is they make these cells from iPS cells, and they try to make the company Fate, which tries to make these cells as a generic cell, which is accepted by everybody. So they would put this modified T cell receptor, which recognizes, let's say, a given cancer, and you can take it off the shelf and it would be acceptable to many patients. So this is all under development. And I believe that's maybe realistic.
Okay. That leads into another question of once somatic therapy techniques are proven to be effective at combating age-related diseases, do you see a path of scaling this technology to a point where it's affordable?
Yeah, so that's a very important question. So when iPS cell technology was really became really mainstream, of course, people want to make these patient-specific iPS cells because they get accepted for any disease, age-related or not. But of course, this is almost not practical because it's so expensive. So people like Yamanaka and other people do this as a proof of principle. Yes, it works for some eye diseases, has been done in clinical trial, and now they're preparing for Parkinson's disease. But make it patient-specific. You would have to take a somatic cell from the patient, reprogram it, grow it up, do all the assays for that they are in FDA relevant. It's impossible to pay for that. So the question is, could you make a generic donor cell? And that one approach would be to isolate specific iPS cells. They're called HLA homozygotes. So HLA is really the key gene which leads to rejection. And they're very rare people who are homozygous for the same allele. They may be very rare. And so Yamanaka did, some other groups do this. They went to blood banks in Japan and tried to isolate from blood cells which are homozygous, iPS cells. And they estimate for the common alleles, so common, they would need 15 different cells to treat 70% of all patients, depending on the combination. It's on the combination of HLA. For the more rare ones, you need many, many more. So these would be normal cells, and they could be just off the shelf and used. This, if you have those, that could be a solution.
Would you say that's one of the biggest, what are the other barriers to?
Well, another approach would be to make the iPS cells invisible to the immune system, right? So what you could do is you could inactivate the HLA on the antigens which lead to rejection. People do this. It's somewhat dangerous because if these cells are invisible to the immune system, they could make cancer and the immune system couldn't fight it. So, this is a double-edged sword. People still do that, try to do this, make them invisible and put suicide genes in those. So in case there's an accident, cancer grows, you just turn on the suicide gene and kill all the cells. So there are various approaches to adapt this technology to the clinic. And I think we're sort of away from this for direct application.
How far in the future do you think germline correction will become routine, or will it ever become routine?
So that's a big question. So I must say, I personally cannot see any justification to treat embryos which have a mix of embryos, healthy, wild type and normal embryos. Let's say Huntington's disease, the parent wants to have a normal child, right? And it's heterozygous. So 50% of the embryos will be normal, 50% will be affected. I cannot see at all you would manipulate those. For the reason I said, because you would manipulate 50% normal embryos. So you do pre-implantation genetic diagnosis to get the right embryos. This, I think, make it not necessary to do this. So it's really for the very rare cases when you have a homozygous parent or both parents are mutant, and there's no way to get a child which is genetically related and healthy. So these are the very rare circumstances where you, to my opinion, you could consider this if the scientific issues are solved, and using spermatogonia stem cells or iPS cells making into oocytes could be one way of doing it.
So while we're on the still on the ethical side, who is involved in the ethics discussions that are ongoing now that you've
there has been a new committee made by the National Academy of Science and the Wellcome Trust and other institutions. So there's lots of people come together to discuss this. These were patient advocates, these are ethicists, social scientists. It really needs to be consensus. It's a very complex way. The scientific part is one only, and the other parts also have to be discussed. And that is, of course, you need a consensus, and countries will come to probably different conclusions. What is permissible and what is not permissible.
So now that we have CRISPR, base editing, RNAi, modified mRNA, et cetera, what major tools are we missing for control of biological systems? What new tools do we need?
So I give from my personal, what we lacking at the moment is we have to learn better, but this is more technical, how to make really mature neurons, mature muscle cells from ES cells. This is complicated. This would be important, and I think that's a very important issue. But I believe these technologies we have, the embryonic stem cell technology and iPS cell technology, and the CRISPR-Cas-based gene editing are some of the key technologies for modifications. What we now need to understand is how the genes are regulated. It's so complicated. What's the difference between genes between you and me? And the subtle differences which lead then to many differences, many risk factors, as I said before, put you at risk to develop Alzheimer or not. How do we put this together? How do we understand that? And I think that is still pretty much in the beginning to try to unravel that.
Do you have any comments on freezing or saving umbilical cord blood stem cells?
Mm-hmm. Yeah, so that has been very popular. I believe it's a total waste of money. I would never do that. So the reasons are following. It's based on a misconception that you can make out of these umbilical cord cells any cell type. This is nonsense. You can make blood cells out of those, right? So that's the first thing. And if you blood cells, sure, if you have blood cells from the cord blood, you would have enough blood cells for transplantation of a very young child, less than a year. But you don't get your diseases, blood disease, leukemia, so as a 1-year-old, you get it as an adult. No way this would be enough to do this. So it makes no sense, it's very expensive. So companies make money with this. And having the iPS technology, and you can do it from the adult. It's no problem. So I think it would be misconceived to do this. I think it's a waste of money.
Okay. Just a good general question, I think, from Preston Ford. What book or books would you recommend as an introduction to the subject?
So Jennifer Doudna from Berkeley, she is one of the key people who developed the CRISPR-Cas technology. She wrote a book on this. I don't remember the title of the book, but I think it's very worthwhile to read. It gives you really the personal involvement of her and gives you quite a lot of information, what the technology is and what. So I think I would recommend that book.
Okay. Yeah, I don't know if I haven't read her book, but I don't know if she covers the ethical issues or the exploration.
Ethical part, everything. Oh yeah. No, that's very important in this. Yeah.
Okay. It might be a crack in creation, gene editing, and the unthinkable power to control evolution. Is that it?
Yes.
Or?
Yes. So they're the transhumans, right? The transhumans think we have to do that. We have to do enhancement. We have to change our genome. We cannot wait for evolution. It's true, and of course, probably our progress or whatever you want to call it in humanity is very rapid. So they would argue That's our only chance to survive. We probably will live in a, if you go on space travel to escape the Earth, which explodes or heats up, we might need humans to consume less oxygen. We have to modify them and whatever, right? So those are people who would absolutely say we have to do everything. So yes, one could probably push evolution of species and do it using this technology. Could you do this? Should you do this with humans? Well, that's a complex question.
Yeah. Okay. I think I'll just one last. I think we have gotten through all of the questions. Amazing.
Okay.
There, and yeah, so we're just. Yeah, does anybody else have any questions out there? Anyone? No. Okay. Yeah, that was very very interesting. Definitely a lot to yeah, a lot to think about. A lot to, especially with yeah, maybe someday we will all have clinics where we can just go in and have them draw blood one day and then come back in a month or so and get another injection and they'll cure the cancer. That would be.
Yes. Yes. No, I think it will develop very rapidly.
Yeah, I mean, I was just thinking when I started, this was I started in biology in your class. This was all complete and total science fiction.
Yes.
Gene editing, and now it's becoming routine in the lab setting with mice, and probably by the end of maybe by the end of the next decade, it will be routine on some level in humans.
Yeah. That's possible.
Yeah, so that would be so will have to be another request for the name of the book and the author. So it's Jennifer.
Don't know. Mm-hmm.
Okay. The author. Okay, if you've got the author, should be able to find that. Okay, I think if there are no more questions, I think I'll just say if there's any last comments, or we can just go ahead and bring things to an end. So.
Okay. Okay. Well, thank you.
Thank you very much on behalf of the MIT Club of Boston. It's been really fascinating, and just a reminder to everyone: we will be making a recording of this session available on the club's YouTube channel in the near future.
Okay.
Okay. Thank you.
Bye-bye.
Bye-bye.